Hi everybody, Alex Zhavoronkov here. Today I'm here in Hong Kong together with Dr. Frank Pon, who is heading our Hong Kong operations and is also responsible for PandaOmics and associated ecosystem. For those of you who don't know, over the past 1.5 years, Insilico's main focus in AI was on benchmarking. So we benchmark everything, even human performance, but of course we like to benchmark machine performance in every area of drug discovery, from target ID, disease modeling, small molecule chemistry, biologics, clinical trial outcome prediction, so everything. And since at Insilico we have now 31 developmental candidates, we can now utilize those programs that have never been licensed to anybody as a program as a benchmark. I think I coined this term. And we have, from every developmental candidate, we get about 1.2 thousand benchmarks from start to developmental candidate nomination. So one of the most important benchmarks is in target discovery, target retrieval, disease modeling. And today with Frank, I'd like to talk a little bit about our new TargetPro platform and also the benchmarking initiative that he and his team has developed. So let's go. What's TargetPro?
TargetPro is an advanced target discovery model. So basically, it is trained with the benchmark for all the current target discovery platform, and specifically to each of the model performance. For example, like in the past, we have many omics data models and also the text-based models. So how well are those models being performed in terms of the novel target discoveries in the, let's say, the past 10 to 20 years? So, and then we develop benchmark. So this is also our focus, because once we have the benchmark and then we know what is the best and what is the right directions, and then we can train our models to increase the model's performance in that directions. So this is, first we have the benchmark, and then based on the benchmarking, we develop our TargetPro model. And then we not only limited our benchmarking with the only the target discovery platform, but also many of the frontier LLM models. And after our training, our TargetPro model can outperform all these platforms, including the LLM models.
So currently in target benchmarking with our target bench, how well do the frontier LLMs perform? And I'm talking about, ChatGPT 5.5, Opus 4.8, and others. Where are they compared to specialist target discovery tools?
Well, I think at the moment they perform quite well, okay? But of course, there are many areas that we can improve, especially for some targets, let's say the druggability, safety areas that I think our TargetPro models has performed much better.
Let me ask you that differently. How badly do they suck compared to state of art? So is it like 10%, 20%, 30%, if you are looking at the target bench comparative performance score?
I would say overall, I think it is about 10 to 20%. It depends on the disease area.
So basically, you are saying that the frontier models like 5.5, 4.8, and of course, we're waiting for the mythos to be released, they are only 10 to 15% worse than our current best-in-class tools?
I think overall, yeah, it is. Yeah. But I think what more important is, let's say we have our benchmarking, and then with our MMAI Gems, okay, and then we can, for example, we take the Alibaba's Qwen, and then we train it, and then it can immediately perform the best, okay, among all these LLM models.
But the best, that means like 10 to 15% better than frontier model, or more than 10 to 15%?
More than, yeah.
So basically, right now, what we've seen with our MMAI Gem is a platform that allows you to post-train and fine-tune your LLM. It could be a very frontier LLM, or it could be an open source LLM like Qwen 3.5 of different variable size and density and architecture. We can post-train it on our data and also use some of our models as teachers. So when you fine-tune some of the Qwen models, we see that they outperform in a pretty dramatic way the frontier LLMs like ChatGPT 5.5 and Opus 4.8, and we expect to continue outperforming in multiple tasks because for some of the tasks, it's not only about the reasoning or the data, it's also about the workflow, how you came up with the target. So frontier LLMs, I don't think they can still get to that point constructing complicated workflows that account for quite a bit of tacit knowledge that is encoded in the minds of biologists and experienced drug hunters who have done this before. And also just very accurate historical data in terms of even clinical trial outcomes. Very often you don't know if it succeeded or failed because clinical trials.gov may not tell you that. You also need to look at the stock prices of the companies and really understand whether the target worked or not. And of course, going back to the preclinical testing, it requires quite a bit of experimental evidence. So we do have that. We actually have one novel target that has never been in the clinic, just passing phase 2A study with very promising safety and efficacy, planning to go phase 3, and many others. So that comes from the platform. We have not seen that with any LLM yet. However, after post-training in our MMAI Gem, you probably would be able to acquire the same level of performance as our PandaOmics platform. It doesn't mean you don't need to buy PandaOmics anymore, but it definitely gives you a competitive edge. Speaking of PandaOmics, Frank, what are the new features? Where is it going? How is the platform selling? And what superpowers does it give to the user after? When did you take over as product manager? How many months ago?
It should be 7 months only. Yeah.
Yeah. So in 7 months, what did you do?
Well, I think we made the biggest updates or advancement of the PandaOmics since the product launch. So in terms of the PandaOmics, we added the fully support of the single cell data analysis. We have the pseudoblock to fully connect it with the target ID. So it is very important because we are not just talking about a standalone single cell analysis that people can do, but to connect the single cell analysis with the target ID that is how we identify TNIK as the best target for IPF. Because if we only look at the average or the overall about RNA-seq, we cannot know that TNIK is actually very upregulated in the myofibrils of the IPF patients. So with PandaOmics now 6.0, you can easily replicate our findings for the TNIK. Yes, and you have the potentials to identify the next novel targets. Yeah.
How well is it now propagating, and do we get significantly more users for PandaOmics?
Yes, I think overall it is good. Yeah.
Great answer. Not premeditated.